So we will now get deeper into things that we already know, but we will have a new insight into each one of these topics. So we will divide the lecture into multiple sections. Each section will have its own video, so they will not be long. So we will start by revisiting the concept of experimental unit. So experimental units are the smallest division of the experimental material such that any two experimental units can receive different treatments. And each experimental unit gives a single observation. And we already know that if we have multiple technical replicates, what we have to do is to take those technical replicates, compute the average of them, and then that will be the observation for the experimental unit. So a clinical, let's see an example. So a clinical trial for a new ointment is designed as follows. So there will be three groups. The group 1 uses the new gel for 12 months. Group 2 uses the first new gel for 6 months and a placebo for the next 6 months. And group 3 uses first the placebo for 6 months and the new gel for 6 months. So the experimental unit are not the people in the experiment, but the period of 6 months of each person, because we can change the treatment at that level. We're assuming this is a crossover design, and we're assuming that using an ointment during the first period will not affect the nature of the patient for the next treatment. So in agricultural crop trials, the experimental unit cannot be each plant, but it is normally each plot, because so plots must be large enough to be representative of large fields and remove the interplant variability, and they have to be small enough to be manageable and remove variability between soil differences. So if there are gradients, then they should be identified. And typically in this kind of plots, long thin plots are preferable. Let's see another example. So trees are normally treated independently. So each tree is an experimental unit, and in a clinical trial, every patient is given a new drug or the best current treatment or placebo. So the patient is the experimental unit. The same happens if you inject your treatment to mice. So the mouse, the animal, is the experimental unit. There are many experiments in which the treatment is on the foot of the cage. So if you have a pen or cage, then each animal cannot be given a different treatment, but you are giving the same treatment to all the animals in the cage or in the pen. So the whole group is the experimental unit, and this is called cluster randomization. So the whole pen is randomly assigned to the treatment or not. And let's say that you're doing an experiment in which you are testing new educational systems or new ways of teaching. So all the children within a class is the experimental unit, because children independently cannot receive a different treatment. So if you are teaching the class in some specific way, the whole class is the experimental unit, and if you need a larger sample size, what you need is more classes. And this is an interesting example, because the experimental unit depends on what you are considering. So consider a study that is designed to test a parenting intervention. So you give classes to the parents, but you measure the behavior of children at home. So the parenting program teaches parents specific behaviors through classes between 6 and 10 parents and 2 trainers. Assume that the content of the classes, the specific behaviors taught to parents, have been chosen through a program of research that has previously shown their efficacy with individual families. So with individual families, they have tested these treatments, and then they want to see if by giving them in classes between 6 and 10 parents, they also have some effect on the behavior of children at home. So do changes to a child's environment through changes in his or her parents' behaviors result in improved child behavior at home? That is your research question. Okay, so this question assumes that the intervention will change the parent behavior, and then you're asking if those parent behaviors are influencing their children. So the family represents the experimental unit. Why? Because the parents are the same for all children within the same household. So you apply the treatment to the experimental unit, that is the parents, and then those parents have multiple measurements that are kind of technical replicates that are each one of the children's. And now there is another question. Does instruction in parenting skills change parents' behaviors at home, assuming that those new parenting behaviors will lead to improved child behavior? So now it is the instruction. You're asking about the instruction. So if you ask about the instruction, then the experimental units are the parent trainers, and then the different parents and the different children within their parents, they are technical replicates of your experiment. So all of them, all these technical replicates would be averaged and applied to the trainers. And here is another example in which, for instance, let's say that you want to detect a rare disease in a population. So you collect blood samples, they are pooled into groups, and then the pooled sample is tested for the disease. If you cannot find it, none of the individuals in the pool has the disease. So if you compare different analysis of this kind, then the experimental unit is the pool, because that is where you can change your, let's say, your analysis technique. That would be the treatment in that case. And yeah, once you find the disease, then you would open the pool and make comparisons for each one of the individuals. So pooling is a very effective way of cutting down costs. And if you change, for instance, the analytical technique and you want to see if there are differences in their different sensitivities, then the experimental unit is the pool. Using parts of a whole as experimental units reduces the variability of the measurements. So for instance, 2 leaves from the same tree may receive 2 different fungicide treatments. So the leaves from the same tree, they share all the genetic background. So the response to the fungicide for the fungicide will be less variable in these 2 leaves, but you have to make sure that you try sufficient trees, because otherwise your results only generalize to that tree. So this is an example in which you try to minimize the variability of the observations by using match pairs, but you need sufficiently large number of large pairs. So for instance, 2 fruits from the same tree receive 2 different storage treatments, or different parts of the same bake mix receive different cooking treatments, or each eye of the same person may receive different eye drops. So all these are match or pair data, and the observations are the differences between the two observations, and there is a single experimental unit, that is the tree here, the tree, the baked mix, or the person. There is a single experimental unit with two treatments, and the true observation is the difference between those two treatments. And also we have already seen crossover designs. So in crossover designs, the population is randomized to one of two sequences. So you have the intervention that you are interested in, so that may be a new gel, and you apply the new gel for 6 months, and then you leave out a washout period so that there is no carryover of the effects of this group into this group, and the other half of the people will receive the opposite sequence. And it is assumed that the first intervention doesn't change the person, so that the measurements that you get in here, they really correspond to the same. It wouldn't matter if you do one of the studies before or after, so it wouldn't matter the sequence. Anyway, that there will be a specific section on crossover designs. When you use crossover designs, you have to be aware that there are some people that do not like crossover designs for a number of reasons that you must be aware of. So for instance, a trial in which a patient receives a sequence. So in red we have the arguments against, and in green we have the questions that we should ask, or what we should guarantee to counteract this possible problem. So a trial in which a patient receives a sequence of different drugs cannot provide legitimate information for future treatment in which a patient will receive a single drug. So here the key question is whether the washout period reverts to the initial condition. So if the first treatment changes the nature of the patient, then this argument is true. Once a patient has received one drug, the patient is changed, it is the same one, and therefore does not provide the same condition for the second drug. And treating a patient with time cocktail of drugs, so the patient will receive two drugs, one first and second one, may provide a more dangerous situation for the patient, for which the dangers are difficult to predict. So we must be aware of possible interactions between drugs. So having given the first one, maybe the second one, there is not a sufficiently large washout period, and then the second one interacts with the first one, and it causes some problems. And the order in which treatments are presented may affect the apparent benefits of the different treatments. So that is why the order of treatments must be randomized from patient to patient. It may be difficult to prevent the patient making judgments about which treatment she is receiving at a particular time, and this may bias the results, whether or not these judgments are correct. So it is better to base your analysis on objective measurements, so you can measure the concentration of whatever in blood. That is objective. It is not asking the patient how she feels. And then you have also experimental repeated measure designs. So repeated measures means that you apply the treatment once, but you measure multiple times at different time points. And the experimental unit is the subject, that is where you have changed your treatment. And the different measures, they are not technical replicates, because you are measuring at different times and the response must be maybe different. But these measures different time points are not independent. So the experimental unit is the treatment, and this kind of designs, they are analyzed through these split plot designs, and we will not see the equation now. But so this would be all for our second thoughts on experimental units, and in the next video we will think about replication.